I'd like to ask the speakers to come up to the table now so we can have our discussion period and go over some of those very interesting issues that were raised. If people are asking questions, could I ask them also to give their name and affiliation just for the record keepers? Thank you.
Samir Cleve, GRU Cancer Center. So this is really a great session, and I congratulate all of you. Rami, I have a question for you regarding toxicities. And in your experience with the work that you've done on drug development, how much, we had some discussion yesterday, how much preclinical toxicity has been of value? And if so, when and where?
So unfortunately, the preclinical data is it's not very informative, and there's very little that we can do to rely on the preclinical models to inform the toxicity, unless that there's something that's very obvious that will help us eliminate some combinations. But in general, we try not to rely on the preclinical models for safety.
So in general, everything you've done from preclinical, whether in large animals or in small animals, but particularly in large animals, you don't think has been a guide for what can be done?
So it could potentially answer a specific question. So for example, when we were doing novel combinations and there were some concerns about a cytokine release. So those kind of experiments, if you're really answering a very specific question, looking at cytokine levels, it can be done and it can help inform the clinical trials, but still the definitive answer will come.
Angela, are you looking into this for potential approvals in the future, or what are the thoughts?
Yes, I mean, the ultimate clinical model in these therapies is the human, and you can have all sorts of clinical models which are assessed to see how they may predict what will happen. But for instance, if you have targeted therapies, you don't necessarily know whether your animal model actually expresses the receptors for those therapies in the appropriate organs. And one particular one I was thinking of is that the rat didn't express it in the lung, and when it went into the human being, actually got a lot of inflammatory change in the lung. So I think trying to pool that expertise and experience is very important to realize where the pitfalls are. But non-clinical models can be very difficult, and they can be misinterpreted and lead to really quite significant problems when they could go into humans, as was seen in the CD28 Tejanero disaster.
So my question is from Mark. You mentioned product safety and labeling in your talk, and I wonder if you could expand on that.
Sure, let me just take a few words on that. Chain of custody is one of the most important things to keep in mind when you move from a single site to a multi-site endeavor for an individualized product. I mean, it's well taken care of in some applications, blood banking, for instance, but moving into the field of individualized cell therapies, that's one of the most important considerations and one that regulators take most seriously. Establishing that chain of custody all the way from multiple collection centers through manufacturing to multiple distribution centers is one of the challenges that a commercial entity has to take on, moving from phase 1 to commercial.
And could you speak a little bit about labeling?
Labeling is an interesting challenge as well, because how to establish labels and as an example, you don't want to print labels because that sets up a difficult auditing system. You have to take care of each label that's printed so that you don't end up with extra labels and not be able to trace your product. So there's a lot of challenges for commercial labeling that don't exist in an academic environment. Just to give you a sense of what's possible in early clinical trials. We used to manufacture a product and write on the bag the expiration date, which was an hour after we made it, and then we would walk it over to the patient and infuse it. In a commercial setting, you don't do that. You know, you have to obviously keep track of each and every bit of the product. So the way that that's established has to be appropriate for multiple regulatory environments. If you're going to have a global product, it has to be, you have to think about different languages. You have to be aware of how to get the labels to the collection facility in time, and so on. It's quite a endeavor.
Jay Brzezowski, NCI, NIH. I'd like to refer to Jed's talk about physician education, and in particular the immune response or immune response related criteria, or response criteria. I also spoke yesterday and referred to some evidence for the need for using these criteria that you so nicely put together in that 2009 paper. And I think part of the physician education that's really important is not just for the physicians taking care of the patients, but for the whole academic oncology community and oncology community in general to understand that, because I've encountered at least tremendous resistance even within academic settings to veering the slightest from the conventional RESIST criteria in evaluating clinical studies, for example, and in people accepting results based on IR response criteria rather than RESIST criteria. And I think that's going to be important in getting acceptance of these approaches and the validity of the studies and reviewers for journals and so forth and so on. So, you know, do you have any thoughts about how to get across more widely this issue of not being so rigidly adherent to the old RESIST criteria when it comes to immunotherapy?
Thank you, Jay, for the excellent comments. I think that it's an ongoing process. You know, at first the simple answer was going to be use overall survival as the primary endpoint, and everything else, you know, kind of will get integrated into that. But I think very fortunately, because we have so many new agents, overall survival actually becomes a more impractical endpoint in many situations because of just simply how long it takes, fortunately because of how long people are living. So I think we need to think more about reasonable surrogates, so landmark survival endpoints rather than overall survival endpoints are now being spoken of. I think if we can put a little bit more mechanism underlying the immune-related responses to prove what we have hypothesized about why tumors may appear larger, we may be in a better situation to substantiate the importance of these surrogate criteria. You know, one or two efforts in those areas, I see my colleague Hedy Rechak there, who I've spoken of a lot about this, using MRI to its fullest capacity to understand the differences between true tumor growth and tumor growth that is a result of immune infiltration. There are now several efforts underway to try and use immuno-PET type of imaging to really image T cells within the patient so that you can try to integrate information about immune cell trafficking to tumors with tumor growth. I think that once we can put a bit more data behind the observations, this will come more easily.
Hi. I could add one thing to that. So in the community, RESIST criteria not used specifically each lesion is not measured as it would be in a clinical trial, but there is a gestalt about whether or not the entire tumor burden is staying stable or shrinking or progressing. So I can't stress enough that having some surrogate markers of whether or not there is pseudoprogression, how important this is when one makes the decision to continue therapy or not. It's not a trivial issue. In fact, in many cases, sometimes these new lesions can be biopsied. So that puts the patient through another invasive procedure with just the concern about what's going on. So we need to establish new technologies as soon as possible.
Jay, I want to add one other thing that builds on Lee's. Oh, I'm sorry if I jump back in there. The liquid biopsy technology, which is emerging, of looking at circulating cell-free tumor-derived DNA. So in the case of the disease that I know best, melanoma, looking for circulating mutated BRAF DNA as a biomarker. This is something that our group, led by Paul Chapman, is looking at as a way to get beyond radiographic measurements, or add to radiographic measurements to say, well, if the tumors appear larger but the circulating BRAF is dropping, then that should begin to tell you something. And so we're looking at the use of liquid biopsy markers as a complement to imaging to try to again add another dimension.
Hey, Angela.
Going back a little bit to the adverse event reporting and education so that these events are reported. I mentioned in the early access to medicine scheme, there has to be very clear information given to ensure that the physicians are properly educated in how to report those adverse events. And some of those will be in terms of what happens to the tumor and response and so on, as well as the patients themselves, because although these therapies are very promising, as been pointed out in several of the talks, it's the longer term effects, the timing of these adverse events, which is not certain. And by that time, the patient may have escaped a little bit from their oncologist. And so they need very clear instructions. There needs to be a very clear pharmacovigilance plan, which the patients participate in as well as the physicians, so that when they see progression of a tumor and so on, they say, oh yes, well, we know that that happens and this is what you can do.
Hi, I'm Judy Keene from the American Society for Radiation Oncology, ASTRO, and I guess this is more of a comment. I think the talks today were fantastic, especially one about education and the importance of being educating the people who are involved, especially with immunotherapy changing so much and combinations therapies with chemo and radiation also being incorporated. And I wanted to mention that we are working as well with SITC, and they will be at our annual meeting with a presentation and working with our people as well. So I really commend ASTRO, ASCO, and SITC and what they're doing with education, because it's incredibly important.
Hi, Lee Krug with BMS. So I just want to go back to this topic about the IR resist and the topic of pseudoprogression, and just to point out that I think these efforts to try to identify ways of eliciting when a patient's tumor is growing and it's related to the immune response, as opposed to situations where the tumor is just truly progressing, are really important, because as was pointed out, this concept of pseudoprogression occurs in a smaller percentage of patients, and it may vary by tumor type. So it may be much more common, for example, in melanoma or renal cell carcinoma, may be less common for patients with lung cancer. And for patients with lung cancer, delaying their another treatment, may result in some decline in their performance status, so that they wouldn't be able to get other therapy. So I think there should be at least a little bit of caution about suggesting that this concept of pseudoprogression is really widespread, and that we need to really work hard on these efforts that were pointed out to try to identify when those are the cases. And use of symptoms, patients, how they're feeling, can be a big part of that. If the patient's tumor is growing, but yet they feel like dramatically improved symptomatically, then that would be a good indicator that maybe the scans don't really reflect what's going on with the disease.
Any comment on that from the panel?
So I think for the immune-related response criteria, it's a two-step approach. So what we've learned over the past 10 years is that there's a need for a different criteria to assess whether patients are benefiting from treatment or not. So the work that Jed and others did really, I think, took us to that place where now even in the FDA guidance document for vaccine development, they acknowledge that there's a need for a new response criteria. They don't specify what is that response criteria, but now with the wealth of data that we have, if you look at all the studies that were done with immunotherapies, all of them had an exploratory endpoint that just tries to characterize what's the immune response criteria or what's the immune response rate that was observed. So now we're at the point where we have to start validating. So we have to standardize, again, what's our definition of an immune responder, and how can we generate data that will be convincing to regulators that this is an endpoint that can be used in clinical trials when we are really assessing the clinical activity. So I think we've advanced our understanding, but now we are at the point where we have to really validate those endpoints. We have to standardize them and then start to use them, at least in the clinical trials.
Okay, question over there.
Lee Newcomer, United Healthcare. Lee, I was struck by your story of the patient who was obviously progressing radiographically, but felt better. And so you were asking a clinical question. It reminded me of a story in New York where I have a friend who fell off her bicycle, hurt her shoulder, came in to see her internist, who took her history, but then ordered an MRI and sent her to the orthopedist. And as she finished all that and went into the orthopedist's office wearing a kind of an open cut top, he said, well, why are we x-raying your broken clavicle? Why are we x-raying your shoulder when you have a broken clavicle? So it's all back to clinical medicine again. And I'm wondering, are we just not being patient enough? Should we simply, if the patient is improving, that's enough of a signal to say we should wait a longer period of time before we search for radiographic evidence? Or is that not a consistent enough finding in these patients that you can't rely on it?
Thanks. I think it's a great.
I was giving it to Lee. I was going to let you off the hook.
He kindly passed it. I think it's a great question, and I don't as much as I want to say that we don't need anything other than our clinical judgment. It is rather infrequent that we see these discontinuities between the imaging and the way that the patient's feeling. It's maybe 10 or 20% of the time. And I think especially when we've been deliberately judicious about not imaging people too often on studies of immune modulating medicines. I mean, it's very unusual to see imaging that's done with any decision attached to it before 12 weeks. Which in the context of the life expectancy of some of these patients is unfortunately a reasonable fraction of that. I think we do need the imaging. I think we just need to be careful about very early time points. That's where I think we can see a higher rate of discordance between potential response and enlargement of lesions on imaging. But I think your point is really well taken that.
And would there be a multivariable for these patients who progress but feel better? Is that a signal to say we should wait on that subgroup?
So what we're trying to do is to try to ferret this out in a prospective way by offering the possibility to patients of continuing past radiographic progression as long as the performance status is maintained on as many clinical trials as possible. And I give a lot of credit to the colleagues from pharma for being open-minded to this, because obviously it does escalate costs and delay the readouts. But I think I think as much as patients would it be expected to say, why would you want us to stay on a medicine that's clearly not working? If they're not feeling worse, it does make sense. And so I think we're all learning about this together.
And Jed, I just wanted to make one more comment on that point. I'm David Kaufman from Merck. One of the experience that we've had actually in mismatch repair deficient colorectal cancer is actually quite interesting here in that radiographic responses were relatively slow, but patients actually felt better much earlier than their radiographic images showed changes in general. And so that, it's a small number of patients at this point, But it appears that there may be different patterns in different disease states, and I think we still need to flesh that out.
So just one more comment. So I think there's two aspects just to add. One is that this is a very important conversation with the patient, particularly in lung cancer where they've gotten standard therapies where typically they might be scanned every 6 to 9 weeks. And all of a sudden, you're now telling them we're going to wait, particularly in the second or third line, where the situation, frankly, gets a little bit more desperate. So I think setting expectations with the patients is really critical here. And this really is a shared decision-making type of initiative.
I mean, also from a regulatory point of view, if you have imaging to support or not support your drug, it's very useful. It's a very hard endpoint. The problem is collecting the data reliably to say that actually I feel better, that yes, this may be happening to me, but overall my quality of life has improved. And quite often in the regulatory period, you see that there is an advantage in terms of overall survival or progression-free survival, but you haven't necessarily got that patient quality data to back it up. And so in some ways, having the imaging on its own can not be helpful in the early stages when you're trying to get regulatory approval, because it's a hard endpoint that doesn't look good, whereas the patient actually can provide that endpoint to say that translated into how I feel, it's so much better. So that's a really important point.
Mark Thuray, FDA. Thank you for the excellent presentations this morning. I have a question for Angela. So specifically, you mentioned about providing earlier access of medicines to patients unapproved and approved. I was wondering if you could speak a little bit more about the adaptive licensing program and compare that with conditional, approval, and speak a little bit about the differences between the two approaches.
Yes, it's struggling sometimes when I look at it as to whether there is a real difference between them, but there's an accelerated approval program and a conditional approval program, and now adaptive licensing. And they really are quite similar, but certainly the previous programs, conditional licensing and accelerated, weren't really being utilized very much. Only 1 or 2 drugs per year were being licensed on that basis. And the difference with adaptive licensing is that at a very early stage, after the phase 1 data have come through and the drug looks promising, is to get everybody together. So that will be, as I mentioned, it will be the EMA who actually delivers the scheme, plus stakeholders, manufacturer, the HTAs and so on, to say what is the best way, what are the best trial designs, how can we get the patients on board, how much data do we need, what do we need to collect in order to get preliminary data to say that this drug in this particular area of high need, we're prepared. The benefit risk will be positive, but a lot of the data, there will be a great deal of uncertainty, particularly about the safety issues, not least because as you have said, that they may come on later. So adaptive licensing will be for the highest risk, most likely to benefit group, a small group, but with very clear criteria for risk management and for pharmacovigilance. And the drug then will be licensed and will then go out and to be used in the real world use, but with very careful monitoring and often registration of the patients. Then those data on safety and confirming efficacy can be collected more easily and more quickly. And if they are confirmed, then that license will continue. Conditional licensing, as far as I understand it, is that the license continues on the condition that you provide the data within a certain time frame, and then the license may be removed. But actually, they're pretty similar. The EAMS scheme, the Early Access to Medicine scheme, which is a UK scheme, is designed to be complementary to the adaptive licensing, so that in a way you apply for both together. So while you're waiting for the marketing authorization, the drug has shown at the end of its phase 3 trials to be actually looking promising. But then you apply on an individual basis for each patient, but it comes with this education pack and so on, so that you get as much information about safety and so on as possible.
So I guess just to clarify, so FDA has a program called Breakthrough Therapy, designation. So the adaptive licensing sounds very similar in some ways in terms of intensive guidance. So at what point, so adaptive licensing includes that intensive guidance, but is that product licensed once approved by that program? Or is it may still follow a bunch of different interactions to get the evidence to provide that initial licensing? I guess I'm trying to figure out at what point is that if approved for that program, is it licensed at that point?
It's licensed at that point, but with a very restricted indication. So it may be it's licensed in third-line therapy rather than first-line therapy or second-line therapy. But if it's used and it's showing its worth and its safety profile with the extra information is favorable, then the indication may roll out to second indication, or even first line, and that's where the adaptive licensing should be helpful. It is still very early, but that's the idea.
If I may, Brusovsky, NIH. To follow up a point that Angela just made, that is about the risk-benefit ratio and adaptive licensing for patients at the greatest risk. There's some evidence to suggest that immunotherapy may work best in patients with more minimal disease. And if that's true, and it's somewhat controversial, but if that's true, then applying it only to patients at greatest risk who have failed all other therapies and are terminal or something like that would maybe miss opportunities to see efficacy in patients with less disease. So I wonder how you deal with that from a regulatory point of view, because you have to look at the benefit side as well as the risk side, right?
I think under those circumstances, you would wait for the result of more formal clinical trials. You could, the clinical trials would take longer, but you could include patients who are at less risk. So you would get more information on safety before you had actually got its license, if that makes sense to you.
George Weiner from Iowa. I appreciate the discussion of the educational programs. One audience we really need to educate is the payers as well, because it's not uncommon now for there to be pseudo-progression and for the payers then to want to pull the plug on supporting the drug. So I think the education efforts need to be geared towards that group as well.
That's an important point. Thank you.
So I would like to thank the panel. That was a great discussion. For the next one, which we're going to be having also another panel with 7 people representing some of what George just mentioned now. We will have small presentations and discussion. So I would like to ask everyone, if possible, to come back at 10:10, and we can start 5 minutes earlier because we're really having a longer type of discussion. And again, thank you very much for all the speakers. You've done a great job. And Malcolm.